Well, good evening, everyone. Welcome to today's live session of week 2 of Immunology course of NPTEL. So today we will be discussing mostly about the responses of innate and adaptive immunity. So I hope you have all gone through the week 2 content from the website, and you have seen that the professor is discussing mostly the responses, the kind of responses that is generated in innate as well as adaptive immune system. So we will be going through those processes once again for a brief summary, and then we will be discussing some problems. And today I am going to discuss some of the questions that are asked in the blog, like the NPTEL blog Immunology. I had gone through it and I found some very interesting questions in this blog. So, okay, everyone, let's begin. All right, so in last week's session, we were going through this innate and adaptive immunity, these two terms. And as we have actually discussed, that innate immunity is something which is the first response that is mounted when there is an infection. When suppose a pathogen probably enters into our system, the first response that is generated is the innate immune response. And the innate immune response, unlike the adaptive immune response, is not so specific, right? This also we had discussed. And if you remember, if some of you were there in my previous session, you would have remembered that we were discussing some PAMPs, some PRRs, and some of the innate immune cells also, like the macrophage, the dendritic cells. These are some of the most important cells of the innate immune system. So for a brief summary, you can see here that when suppose this is a nail that if it suppose pierces in your skin, then what kind of response begins? And if this nail perhaps gets into your skin, then some pathogens might also breach this anatomical barrier, right? And then someone asked, like, whether the mucosal surfaces are also the anatomical barriers. Yes, they are the anatomical barriers. Now what happens, these kind of so these are the pathogens. These are the pathogens that also enter through this piercing of the nail. And then what happens, one of the first cells, one of the first and the most the cell which responds in most number in this kind of a situation are the neutrophils, right? And the neutrophils reside in our bloodstream, right? So they are streaming in our blood, and these neutrophils has to be then so this infection is not occurring in our bloodstream, right? So this infection is occurring in our in some of our tissue, and then these neutrophils that therefore need to migrate to the site of action or to the site of infection, where they will perform its dedicated function. So for these neutrophils to migrate to the site of action, there's a process which takes place which is called extravasation, which is nothing but the crossing of the blood, the endothelial layer of the blood vessels by the neutrophils to come to the site of the action. Now some of the important events that occur here is that so there are many APCs like I had discussed in my previous session also, APCs are what? Antigen presenting cells, right? And these APCs circulate in the tissue, so they reside in the tissue, and one of them is dendritic cell, another one is macrophage, right? Dendritic cell and macrophage. So dendritic cells, they and also these are the mast cells, which are also important, we will see later on. So one important function which is exhibited by the macrophages is that the macrophages bear these receptors called the pathogen recognition receptor, the pathogen recognition receptor, or the PRR. These are borne by the macrophages on the surface of the macrophages, and they recognize what? They recognize PAMs and also DAMs. What are these PAMs and DAMs? So PAM are pathogen associated molecular pattern or pathogen associated membrane patterns. So what are these patterns? So these are particular patterns on the surface of the pathogen, on the surface of the pathogenic organism, which can interact with these recognition receptors, or these recognition receptors can recognize these pathogen associated molecular patterns and damage associated molecular pattern. So these are exhibited by the pathogenic organism, and then what happens? These so this is one kind of someone was asking me in my previous session that what is a TLR? Is it similar to TCR? You will see that TLR is a toll-like receptor. TLR is a toll-like receptor, which is actually a kind of pathogen recognition receptor, right? And so there are particular PAMs on the surface of the pathogen which interact with the TLR, and then it initiates a cascade of event. So first this pathogen is internalized, so it is phagocytosed. So this phagocytosis occurs here. It is phagocytosed, and then it fuses with the lysosome to form the phagolysosome, and then the pathogen is chopped off inside the phagolysosome, inside the lysosome, by the enzymes which are the hydrolytic enzymes which are present in the lysosome. And then these what happens is that these chopped off parts of the microbe or the pathogen is then represented on the surface of the macrophage, which is the antigen presentation part of the antigen presentation role of the macrophages, right? So the macrophage not only phagocytoses the pathogen by recognizing with these PRRs, but also it can present this antigen, these chopped up antigens, to other types of cells, and by which they interact with these other types of cells, right? So now জলদি চলে যাও okay. So now what happens, these neutrophils, they now need to migrate towards the site of action, and that occurs in this process like this. Here you can see the blood vessel, right? Here you can see the blood vessel, and you can see that these neutrophils, they come to the site of action, and one important mediator which helps in this extravasation process are the inflammatory molecules. Okay, so the mast cell, The mast cell, if you remember the diagram of the mast cell, it contains many granules in the cytoplasm, and it releases these granules upon activation. And what happens is that when mast cells release these histamine granules, for example, they increase the vascular permeability. So this vascular permeability is increased by the release of these inflammatory molecules like histamine. There are other inflammatory molecules which we will discuss later on. And then because of this, the permeability of the blood vessel increases, because of which the neutrophils can now migrate to the site of action. Alright, so this is a very brief discussion about what happens in this innate immune response. Now someone asked this question in the forum, that what is there any okay, extravasation I'll discuss. Just give me some minutes. In one of my next slides, you will I will actually go through it. Okay, please wait. Alright, so someone was asking if there is a memory of the innate immune system as well. Like, we are very familiar with the adaptive immune memory, which are exhibited by memory T and memory B cells, Now there is a question that someone asked, that whether there is a memory of the innate immune system as well, and the answer is yes. So previously it was not known. It is a very recent research that is actually also going on right now. That is this is also known as trained immunity. Okay, innate immune memory is also known as trained immunity, and it actually describes the long-term functional reprogramming of the innate immune cells, which is evoked by some exogenous or endogenous insults. So suppose there has been a previous infection of a particular pathogen, and due to this previous infection, there has been a reprogramming of the innate immune cells. Which means that the innate immune cells are now reprogrammed to recognize this particular pathogen, because it has infected previously also. And then because of this recognition, or because of this previous memory, what happens is that it leads to an altered response towards a second challenge. Like, suppose this pathogen is reinfecting, or it is infecting again, then this innate immune cells recognize it, they remember this pathogen, and because of which it happens that they, in the second challenge, in the second infection, there is a secondary response that is generated, right? So when we will discuss the adaptive immune memory, then also you will see that when there is a secondary infection, like when the infection is occurring for the second time, then the immune system reacts completely differently than when it is occurring the first time. And in case of innate immune system also, there has been some of the studies where it was observed that the secondary response to a subsequent non-specific stimulus also can be altered in a way that the cells respond more or less strongly than to the primary response. So the secondary response can be either stronger or can be weaker than the primary response, right? So this is more or less known as the innate immune memory, or the trained immunity. And how is it different from the adaptive immune memory that is actually exhibited by the T and B lymphocytes? So here you will see that this innate immune memory is a very common concept in, like, it is observed in plants, invertebrates and also vertebrates. And there has been several differences between the innate immune memory and the adaptive immune memory. So if you see here, the first one is lack of gene rearrangements. So one of the significant events which occur in the adaptive immune memory is that the gene rearrangements occur which actually produces more competent or more efficient B cell receptors or T cell receptors, which is able to fight the pathogen in a better way. And so this kind of gene rearrangements does not occur in immune memory, or so far it has not been observed. And there has been no epigenetic reprogramming that takes place in innate immune memory. Also, the types of cells are involved, like I told you, innate cells or the T and B lymphocytes. The innate cells are involved in innate immune memory, and the T and B lymphocytes are involved in the adaptive immune memory. And also the receptors which are responsible. So in case of adaptive immune memory, it is obviously T cell and B cell receptors. Whereas in case of this innate immune memory, I told you the receptors are mostly pattern recognition receptors, right? So these are not very specific to the antigen, but they have some kind of specificity. Innate immune memory, yes, the innate cells, the cells that I was talking about, the macrophages, the dendritic cells, the neutrophils, these kind of cells are involved in innate immune system. These cells are only involved in innate immune memory, right? So like the immune cells adjust themselves in a certain way when they face the antigen or the pathogen for the second or the third time, right? So that is what is supposed to be innate immune memory. And there are very interesting articles. So these are some of the articles that you may go through if you find more interest in this subject. All right, so that's about the innate immune system. And somebody was asking me what is extravasation. So let's look at it. So there are a few steps which occur in extravasation. Number one, you can see here, number one is rolling, number two activation, number three adhesion, and number four transendothelial migration. So what is this? Let's look at it. So first what happens, these are the endothelial cells, or known as the high endothelial venules, HEV. High endothelial venules, right? So this line the blood vessel, and the neutrophils, these are the neutrophils which are present inside the blood vessel, right? Now what happens if the neutrophil has to surpass this barrier? So what will happen is that the first step which occurs is rolling. So neutrophils actually roll along the surface of the high endothelial venule, like that. How does this rolling take place? So if you look here, so suppose this is the high endothelial venule cell and this is the neutrophil. Okay, and there is one interaction which takes place if the neutrophil has to roll along the surface of the high endothelial venule, that is mucin-selectin interaction or mucin-selectin binding. So mucins are some cell membrane proteins which are present on the surface of the neutrophils, whereas selectins are present on the surface of these high endothelial venule cells. And once the mucins and selectins bind to each other, then what happens? Then the neutrophils can actually, this binding actually allows the neutrophil to roll along the high endothelial venule. So that is the first step which occurs, that is rolling of the neutrophils. Now the next step is activation. The next step is activation, and activation takes place by the action of one interleukin which is called IL-8. So this IL-8 is actually produced by the high endothelial venule, and then this interleukin or the cytokine actually binds to one of its receptor present on the neutrophil. So this binding takes place, right? So this receptor, so this IL-8 or this interleukin binds to this receptor on the neutrophil surface, which actually allows the rearrangement of the integrin molecules. What are these integrins? These integrins are also cell membrane molecules, right? These are cell membrane proteins attached to the cell membrane, or you can say transmembrane proteins. So it allows the rearrangement of the integrins, and what happens because of this rearrangement is that these integrins will now be able to bind to cell adhesion molecules, which are called ICAM. So the first step you have seen here, that is rolling, and the second step, which is the binding of the IL-8 to the IL-8 receptor, is the activation step. This step is activation. Next is adhesion. So this adhesion actually takes place by the binding of the integrins to the ICAM molecules. By the binding of the integrins to the ICAM molecules, and this step is known as the adhesion step. So I hope you are being able to understand the first step. In the first step, the neutrophil is actually rolling or on the surface of the high endothelial venule. Then there is activation of the neutrophil by the interaction of the IL-8 and the IL-8 receptor. Then there are cell surface molecules like the integrins and the ICAMs, which are now binding close to each other. So which is the process by which the neutrophils are now adhered onto the high endothelial venule. So that is adhesion. And the last step is the transendothelial migration. So in this transendothelial migration, you can see that this neutrophil is going outside, right? So this process by which the this total process by which the neutrophils are going outside the blood vessels are called the extravasation, right? So it is going outside of the vasculature, right? So this is extravasation. Events of endothelial transmigration and the process of extravasation is same. Exactly. Endothelial transmigration and the extravasation, these are just two different terms to actually talk about the same event that is occurring here. All right. So we have just described what are the types of interactions which occur between the neutrophil and the high endothelial venules. Now the macrophage, which actually phagocytoses the antigen and then presents this antigen onto the surface, also produce certain chemicals which are called the cytokines. Right? This is produced by the macrophage, and there are several types of cytokines which perform several types of functions, right? And for example, IL-1, IL-6, TNF-alpha, these are different types of cytokines. When we will learn about cytokines, we will learn in more detail. And it actually elicits different types of responses, like increase in temperature, activation of the T or B lymphocytes, production of the complement proteins, and the production of more and more neutrophils. These kind of different functions are exhibited by these activated macrophages. Okay, and one more thing that I was actually previously telling you also, that mast cells become activated by the interaction of several complement proteins, like these C3A, C5A. These are several complement proteins. I hope you have, so these complement proteins are also produced by the activation of different cytokines, right? And these complement proteins, when they bind to the mast cell, so what happens is that they produce, they release several granules, and these granules are usually inflammatory granules like histamine, prostaglandins, leukotrienes, right? So what is the function of these inflammatory molecules? they help in this extravasation process, right? So they help in increasing the vascular permeability. So the permeability of the vasculature increases, because of which more and more neutrophils can come to the site of the infection, right? So these are some of the inflammatory molecules, prostaglandin, leukotrienes, and the precursor of these molecules is arachidonic acid. If you have if some of you have actually gone through fatty acid metabolism or lipid metabolism, you might have heard about this arachidonic acid, which is a fatty acid, which is the precursor of these kind of molecules, inflammatory molecules. And now what happens, because of this release of these inflammatory molecules, it also generates an inflammatory response. So suppose this nail, if it is pierced into your skin, after some time you will see that there will be redness, there will be swelling on your skin, and there will be pain and heat also. So this is the characteristic of, these are the characteristics of inflammation, right? We have also discussed this in our previous session. And these inflammatory responses are now brought about. We now know that which are the molecules which brings about the inflammatory response, right? I hope I'm being clear to most of you. If you have any queries, you may ask. You may also speak to me directly instead of writing in the message. No problem. Okay, so next I will proceed to one other question, which was okay, thank you, Sindhuja. Okay, now I will proceed to one other question, which was asked by, I think, Shayon in the blog, which is how the dendritic cells present their antigen. Shayon, if you are there, you may also talk to me. Okay, so he was actually asking that in our previous session also we talked about this, that the TCR or the T cell receptor can interact with the antigen only with the help of MHC molecules, right? So MHC is the mediator between the interaction of TCR and the antigen, right? This was what we were discussing previously. Now, Shayon, I think, was asking that how these dendritic cells present their antigen to the T cells, and whether these dendritic cells have MHC also. So yes, the answer is yes. Dendritic cells also have MHC. Any antigen cannot interact with T cell receptor without MHC molecules. Okay, so MHC is very critical in the interaction of TCR with the antigen, or the T cell receptor with the antigen, and that is also exactly the same thing that happens in case of dendritic cells. The antigens, when they present these antigens to the naive T cells, so they also do the same thing via the MHC molecules, you can see here. So the first step that occurs here is that the migratory dendritic cells, these are migratory dendritic cells. These migratory dendritic cells come in contact with the antigens, right? And then they phagocytose the antigen, and then they present it on their surface with the help of these MHC molecules, right? And there are quite complex interactions which occur in between these dendritic cells and some of the bystander cells of the immune system. And then because of this kind of interaction, these licensing of the dendritic cells actually occur. So the dendritic cells now migrate to the lymph nodes. Here you can see the dendritic cells are migrating to the lymph node, and once they migrate to the lymph node, they come in contact with these naive CD4 T cell. I discussed previously that naive CD4 T cells are T cells which have not met the antigen yet, right? So these are naive T cells, and then these migratory dendritic cells present their antigen with the help of these MHC molecules, and then the subsequent activation of these naive T cells take place, right? So this is something that is common between dendritic cells as well as other types of cells which also present their antigen, the other types of antigen presenting cells which also present their antigen via MHC to the T cell receptors. All right. So now we will move on to another concept, which is the cell-mediated adaptive immunity. So I will be having some question discussion with you also, but before that I just want to brush through some of the concepts which were discussed in this week 2. Now in adaptive immunity, actually you know that there are actually two types of adaptive immunity. One is cell-mediated, another is humoral adaptive immunities. What is the difference between these two, we will see. So cell-mediated adaptive immunity is principally delivered by the T cells, okay, and these T cells, they mature where? In the thymus, right? And in the thymus, what happens, these T cell precursors, when they come to the thymus, they are usually double negative cells. So double negative means they do not express CD4 or CD8, any of them. So CD4, CD8, these we also discussed in the previous class, that they are this is cluster of differentiation, and these are some of the co-receptors. So CD4, CD8, these are some of the co-receptors which help in the interaction of the TCR with the MHC. Okay, so these are co-receptors of the T cell receptor. Now what happens to these double negative cells? The TCR rearrangement takes place. So the TCR is rearranged, and now when they express these TCR molecules, they undergo one differentiation where they express both the CD4 and CD8 positive cells. So they are now called DP, or double positive cells, right? And then what happens is these double positive cells now undergo various types of selection. So that happens by the help of these thymic epithelial cells, the thymic epithelial cells. So what do? They present some of the antigens via these MHC1, MHC2 molecules, right? And depending on which molecule of MHC they interact with, now the double positive cells will undergo further differentiation to become single positive. So either they will express CD8 or they will express CD4. So they will become either CD8 positive or CD4 positive naive T cells. Okay, these are naive because they have not met with the antigen yet. They are yet to be matured. All right. Now these T cells, now they will migrate. So they undergo several rounds of differentiation in the thymus, after which they now migrate to the lymph node. They migrate to the lymph node, and in the lymph node there are several zones where the T cells and the B cells they reside. And in the lymph node, two interesting things happens, that is the activation of the T cells happen. And the activation of T cells, either they can be CD8 positive cells or CD4 positive cells, and then these cells are activated. That means they now interact with the antigen and then become activated. When they interact with antigen, they become activated to produce some effector T cells, right? Like Th1, Th2, Th17, Treg, follicular helper cells. So these are some of the effector T cells, and these CD4 plus T cell with the APCs, the antigen presenting cells in the lymph node, they present these antigens. And how does this interaction take place? So this interaction takes place by several molecules. So not only the TCR MHC interaction happens, but also I told you that there is one co-receptor, either CD4 or CD8. So this CD4 here, suppose it actually supports this interaction between TCR and MHC, And then there are other co-stimulatory receptors also. For example, here you can see B7 and CD28. B7 is present on the APC, and CD28 is present on the T cell. CD28 B7 interactions. This is co-stimulatory interaction, which actually strengthens this interaction, and due to these all cascade of interactions, the activation of the CD4 plus or CD8 plus cells take place, and then they generate several memory T cells or effector T cells, right? Now the effector T cells are the T cells which will perform some function. What are these functions? We will also see, but before that, just let's brush up through the humoral part of the adaptive immunity. Like I mentioned, there are two parts of this adaptive immunity, humoral and the cell mediated. And the humoral part of the adaptive immunity is actually what are Th2 and other cells. We will come to this question. Just wait for some minute. Okay, so the humoral adaptive immunity is mediated by the B cells, right? And the B cells are they actually differentiate in the germinal centers. So in the germinal centers, so this germinal center is here zoomed, you can see, and in this germinal center there is dark zone and there is light zone, where different types of B cell maturation processes take place. So first step is activation. Like I talked about the T cells also. The T cells are also need to be activated first by the presentation of the antigen. Here the B cells are activated by the presentation of the antigen by the T helper cells or the TH cells, right? T helper cells. So they present this antigen to the B cell, and because of which, so what happens first, these B cells, they have immunoglobulins on their surface with which they can bind to the antigen molecule, right? Because of the binding of this antigen to the immunoglobulin, they now start to express the MHC class 2 on their surface, the B cells, right? They express MHC class 2, and these MHC class 2 molecules now interact with the T cell receptors of the T helper cells, right? The MHC class 2 T cell receptor interaction starts, and I told you already that there will be co-receptor CD4, and there also will be co-stimulatory interaction between the B7 and CD28, right? There is another interaction which actually is important for this activation, which is CD40 ligand and CD40. So CD40 is expressed by the B cell, whereas CD40L or CD40 ligand is expressed by the T helper cell, and this is also one major component in this activation process, in this interaction between the T helper cell and the B cell. So now once the T helper cell presents this antigen to the B cell via MHC, then this B cell is activated, and now it undergoes the second step, which is somatic hypermutations. So somatic hypermutations are what? They are basically rearrangement of the antibody producing genes or the antibody receptors, the B cell receptors. So why does this hypermutation take place? So they actually in order to develop more efficient antibodies, you understand? So somatic hypermutation takes place in the dark zone of the germinal center, and then when the somatic hypermutation is completed, these centroblasts, so these centroblasts are basically precursors of the activated B cells, now they move on to the light zone of the germinal center, and then this class switching of the first step that occurs in this light zone is that they first undergo affinity maturation. So what is affinity maturation? So suppose all these B cells are producing particular B cell receptors, right? some of them might be very active, some of them might be less efficient, right? So now the B cells also have to undergo a selection process in the slide zone, where the high affinity B cells, the high affinity in the cells expressing high affinity B cell receptors will be selected, and this selection takes place by the help of follicular dendritic cells, which help to select the high affinity B cells. And then finally these B cells undergo class switching. So class switching is nothing but producing different classes of antibodies like IgG, IgM and all these. And then after this class switching, either memory B cells are produced, which will be contributing towards the adaptive immune memory, or the plasma cells are produced. And the plasma cells are what? They will produce these secreted forms of the B cell receptors, which are nothing but the antibodies, right? And these antibodies will then move on to the site of action to bring about several effector functions, right? So what are these effector functions? That also we will go through once, just very briefly. That is, here you can see there are two arms of this adaptive system, humoral and the cell mediated. And in case of cell mediated immunity, you can see that these are the two types of T cells, either the cytotoxic T cell, which produces the CD8 receptor, and the helper T cells, which produce the CD4 receptor. Now the cytotoxic T cells does what? What is the effect of these cytotoxic T cells? They kill the target cell by the action of some enzymes called granzymes and porphyrins. So they actually kill the target cell by the process called apoptosis, in which, so these granzymes and porphyrins, what they do is that they on the surface of the target cell, they produce holes such that the contents of the target cell is released out and the target cell dies. So that is apoptosis. So this is brought about by some Fas-Ligand interaction, which we will learn later on in the later part of this course. And this is actually mediated by the cytotoxic T cells. What is the effect of the helper T cells, which produce the CD4 receptors? So the helper T cells produce one important class of molecules which are called the cytokines. And cytokines are depending on the type of cytokine which is produced, T helper cells are actually differentiated into various types of T helper cells, which you can see here: Th1, Th2, Th17, Treg, T follicular helper cells. So someone was asking, what are these different types of cells? They perform different types of functions, different types of effector functions, right? And they are actually produced by the activation of different cytokines. So in different cytokine environments, different T cells are these particular T cells are differentiated. And suppose these Th1 cells, these Th1 cells, they activate the macrophage. The Th2 cells produce IgE molecules, which help in this antiparasitic immunity. Th17 cells are some of the cells which produce some chemokines. So chemokines are the chemoattractants, right? We were discussing this in the previous session also. Tregs are quite important in this case, because Tregs are the particular cells which suppress other types of T cell response, which will also be required in some cases, we will see. And T follicular helper cells helps in B cell development in the germinal center. So these are the different types of T cells which are produced, and this is the effector function of the cell mediated immunity. And what are the effector function of the humoral immunity? First of all, the antibodies are produced, right? And the antibodies perform three different types of effector functions. This was something we were also discussing in the previous section. That is, they perform either neutralization, opsonization and complement activation. Some of you were also asking me what in the previous session also, that what is the difference between neutralization, opsonization. Neutralization is nothing but directly actually neutralizing the toxin or directly neutralizing the pathogen. this is neutralization, which is mediated by antibodies. So the antibodies coat the pathogen and then directly neutralize it. Or it can also be opsonized, which is mediated by some of the macrophages, right? What do? They so once the antibodies coat the pathogen, these antibodies have these Fc region which are recognized by the Fc receptors, and then they internalize the Fc receptors of the macrophages, and then the macrophages internalize the pathogen, right? Internalize the pathogen and produce and this process is called, and then they destroy the pathogen, and this process is called opsonization. And the other one is complement activation. And complement, there are several complement proteins which are produced in the liver. And what do they do? So these are some of the C2A, C2B, C3A, C3B. These, what are these? We will discuss later. But what do they do is that these complement proteins help in lysis, either lysis of the pathogen, and they form this complex called membrane attack complex, where they cause the lysis of the pathogen by production of this membrane attack complex, and they form a hole in the pathogen and then destroy the pathogen. Or it can also cause opsonization of the pathogen, or it can also enhance inflammation, which are mediated by some of the innate immune cells, right? So innate immune cells, I told you already, produce some inflammatory responses, which can also be, like I told you already, that mast cells are activated by complement proteins. So this is the inflammation part of the complement activation. So this is more or less to this week's content. I hope you are being able to understand what are the different responses in the innate immune system and what are the different responses in the adaptive immune system, and how these innate and adaptive immune cells are maturing in the thymus, or in the T cells or the B cells. They are maturing in the thymus or in the germinal centers of the lymph nodes. And then finally, what are the effects, what are the different effects of these adaptive immune system, the humoral arm of the adaptive immune system and the cell mediated arm of the adaptive immune system. Alright, so now we will quickly go through some of the questions. So please start answering. Germinal center of the lymph node is the region of maturation and activation of which of the following cells? Okay, I'm getting the answers.
Srimay, I have one question.
Yes.
So regarding this thymoid epithelial cell,
Yes.
there when the B cells are getting activated, so it's basically cleaved to MHC molecules, right? The CD receptor. So if it is already attached there, then how later point of time it will attach to any other antigen?
No, actually, this activation process is quite interesting. So suppose one thymic epithelial cell is interacting with one particular cell, the T cells, and then they are actually helping the T cell to get selected. So what is this selection means? This selection actually means that suppose this particular TCR is good enough, right? So you can now go on to kill some pathogens, right? So that is what is selection, right? You are this particular T cell is good, this particular T cell is not. So when this selection happens, when a particular T cell is selected, what happens is it now further differentiates. So this T cell is released from the thymic epithelial cells, and then they undergo further rounds of division and differentiation to produce more and more types of these particular T cells, which will be actually expressing identical T cell receptors. Are you getting my point?
Oh, yeah. So that one may not be necessary, but that identified, so it can copy and generate more for further processing.
Yes, exactly, right.
Okay. Thank you.
Okay. All right. So this is most of you have answered right. This is the answer, the B cells, right? Germinal center. is the region of maturation and activation of the B cells. Alright, moving on to the next question. These are quite straightforward questions. Integrins bind to which of the following adhesion molecules? Right. Yes, I'm getting the answers. Yes. Most of you are correct. So remember, do you remember where these integrins and cell adhesion molecules were there? Where were they? Can someone just bring this context here? Anyone can point out? Extravasation, right, very good. So the integrins are present on the neutrophils. Where are the cell adhesion molecules, which are the, yes, the cell adhesion molecules are present on the HEV cells, and the cell adhesion molecule which interact with integrins or bind to integrins is ICAM. So most of you have given the correct answer. And someone said selectin. Please remember, mucin binds to selectin, not integrins. Okay, the first step that occurs in extravasation, which is the rolling of the neutrophils, there the mucin and selectin binding takes place. Yes. All right. Okay, moving on to our next question, which is not a mediator of inflammation? Histamine, prostaglandins, integrins, or leukotrienes? Okay. All right. All right. Most of you are answering correctly. All right. So that is the correct answer, which most of you are answering is integrins. The rest of the three molecules we already talked about, that these are some of the inflammatory molecules or the mediators of inflammation: histamines, prostaglandins, and leukotrienes. But integrin is not. Integrin is a cell adhesion molecule. Okay, so that is the correct answer. Yes, yes, Shubham, you are right. Remaining three are secreted by the mast cells. Alright, next question. CD40L is present on the surface of which cells? Okay, this is also most of you are answering correctly. One answer was B cells. That is wrong. The right answer is helper T cells. Just going back to that slide once, just to give you one more brief of it. That is, these CD40 ligands are expressed on the T helper cells, right, which help in the help in strengthening the interaction between the TCR of T helper cells and the MHC2 of the B cells, which helps in activation of the B cell in the germinal center. Okay, so CD40 ligand is present on the T helper cells. So that is the correct answer. Okay, moving on to the last question of this session. T cells which express CD8 co-receptors, they differentiate into which types of cells? Well, yes, most of your answers are correct. that is T cells which express this CD8 co-receptors differentiate into cytotoxic T cells, and they kill the target cell by the help of these molecules I already talked about, the granzymes and the perforins, right? And they help in the apoptosis of the target cell. So that is the correct answer. Yes, Shaunak, actually I am sharing all the PPTs with the NPTEL team. So I hope you will get the PPTs as well as these lectures. All these lectures are recorded and they are deposited in YouTube. So there will be links, YouTube links, which will be, I think, shared by the NPTEL team with you guys, so that you can go through these lectures. Even if you are maybe you've missed one of the lectures, you can go through all of the lectures in YouTube. I'll be sharing the links. I'll be sharing the PPTs as well. Yes, please ask. Any of you also, if you have any other question. No problem, you can actually go through all these lectures. Okay, just a minute, a question actually. Disappeared. Just give me a moment. Shubham, can you send me or can you ask the question once more? Okay, CD40 binding and okay, CD40 and CD40 ligand binding, that I'll explain. And you are asking why we have more APC like macrophage, B cell and dendritic cell, and why not only one cell? Okay, that's a very interesting question because these APCs, how do they present antigen? So if you have to present the antigen, you have to first recognize the antigen, right? And you have to first recognize that when some organism is entering into the body, that this organism is harmful, is a pathogen, and there has to be a recognition by the cells of our body that this is a pathogen and I have to fight it. So this recognition occurs through these PRRs, as I have already mentioned, that these PRRs are what? The pathogen recognition receptors. And these receptors are actually recognize different patterns. So there are different molecular patterns all across different pathogens, as you must understand that since there are so many pathogens, right? So of course there will be different patterns on these different pathogens, and these different patterns has to be recognized by some or other pattern recognition receptors, and these pattern recognition receptors are differentially expressed on these APCs. So why are there more APCs? Like why are there macrophages, B cells, and these innumerable numbers of APCs and not only one APC? That is because that there are several receptors which are present on the APCs, which are, so some receptors are expressed by macrophages, some receptors are expressed by the dendritic cells, like that. So because of which there are several there are several APCs. So I hope that answers your question. Yeah, okay. And Shubham was asking CD40 and CD40L binding. So this is nothing but like just how CD28B7 binding also occurs. So these are cell adhesion molecules. So these are some transmembrane proteins which actually interact with each other when the T helper cell is presenting its antigen to the B cell during B cell activation. So this T helper cell is presenting its antigen via the MHC molecule to the TCR, and there is one co-receptor which is the CD4, which also helps in binding to the TCR and the MHC. And there are actually, so this interaction is, like, it occurs in several steps. So there are several limitations, so there are several checkpoints. So if the TCR and MHC is only interacting and no CD40 ligand interaction, or no B7 CD28 interaction takes place, then this B cell activation will not take place, right? So there are several checkpoints here, and in this case, so not only this TCR MHC interaction with the co-receptor CD4 interaction is important, but also this co-stimulation by the CD28 B7 interaction, as well as the binding of the CD40 ligand on the TH cell or the T helper cell to the CD40 receptor on the B cell is also essential for this kind of activation of the T cells. Only when these interactions are complete, then only the B cell activation can take place. All right. Anyone having any other question? All right. If you don't have any other questions, we may end today's session here. It was very good interacting with all of you. And please remember that whenever you have any doubts, please post it in the blog, the blog of the immunology course. And we will be discussing these questions right live in these sessions. Okay. So please keep on posting in the blogs, and you can directly ask me in the sessions also. So bye bye. See you in the next session, this time Tuesday 7 to 8 p.m. All right. Good night.